Hello, everyone. I want to talk about the update on our research in Hernandez lab. I will talk shortly about the experiments that I presented last year, only one graph about the final result, and the two experiments that we've done since last meeting, which are intercanopy lighting and another second cycle propagation. So all the experiments that I've done so far, photoperiod, we also worked on the increasing plant density based on leaf area index, light spectrum, dotted plant size estimation with image analysis and computer vision, and light distribution, top versus intercanopy, and mother plant second cycle propagation. I will focus mainly in the two last ones. In all the experiments, I will not go over all the material and method again, but we always control all the data with data logger about like temperature, relative humidity, CO2, light, everything on the plants. And for the time of harvest, we always harvest them based on their distance from the mother plant. So we have all the data based on like a spatial data of the data plants. For photoperiod, very shortly, we had 3 photoperiods on the cultivar Monterey. And with shortening photoperiod under the same daily light integral, we saw that we can have 18% more daughter plants. We reported this last year. I just added this yesterday after the grower talk. This is the number of flowers that we picked over the mother plant, the cumulative number over the weeks. And we saw that on the higher photoperiod, we have a little higher number of flowers, around like 15 to 20, all of them, but it was not significantly different. We followed up with another spectrum research. We had 4 spectrum, a higher red, lower blue, a lower red, higher blue, and both of them with far red substitution. And again, with the cultivar Monterey, we also, in half of the plants, we picked the flowers over the experiment. For half of them, we did not do the flower removal to also see the effect of that. Per mother plant, we saw that the number of daughter plant per mother plant was not affected by flower picking. So these one are the ones that flower was removed during the growth period. And the right side, the flower was not removed. You can see if we remove the flowers or if we do not remove the flowers, at least in this experiment, we did not see a significant difference on the daughter plant production. Also, there was not an effect of adding forage. You can see per area. So this is like around 30, 35 to $50 plant per mother plant, which under this planting density of 12, it is around $400 to $600 plant per area. Yes, we did not see any flower removal or far red application on the number of produced daughter plants. But with the main two background spectrum, high blue, low red, and low blue, high red, you can see a higher red proportion can increase the number of daughter plant production by 46%, which is also significantly different. You can see the number of flowers per mother plant, and you can see far red addition can significantly increase the number of produced flowers, but no difference between a higher red, lower blue. This one was a little more daughter plants. So we can see there was not actually a trade-off also between the daughter plant, the runner initiation or flower bud initiation between the two main background spectrum.
Well, can you
Yes.
just because can you remind me what the full period was on this?
16 hours. Yeah, it was here. Yeah. So for the period of 16 hours, sorry, I didn't go over all the details because of the time and all the data that I have. So yeah, please stop me if wherever I'm like too like fast or. So we talked, Ricardo and I talked, and we decided to do another experiment to increase the planting density. So at photoperiod, we had 9 plants per square meter, and we wanted to know how much we can push the plants and have a higher planting density because in cultural aspects, if you are a producer and you want to have more daughter plants, like not regarding the environmental condition, with cultural practices, you should probably like keep the plants a little longer to have more daughter plants, and that adds to the ununiformity of the daughter plants, which means the runners are gonna produce more. The daughter plants that are closer to mother plant are gonna get bigger and bigger because they are closer to the source and there are bigger sinks, and your variation will be higher. So we were thinking, what if we have more runners in area instead of like longer runners? So we increased the planting density, and we hypothesized that yes, the number of daughter plants per plant will drop because of the competition, but probably the yield per area will be higher in higher planting density, and the quality of daughter plants will also drop under higher planting density. And our hypothesis was approved, and we saw that under a higher planting density, you can see here, this graph is per mother plant. This is 50% increase and another 50% increase, 100% increase. And you can see the per mother plant, it dropped, per area, it increased. However, as Ricardo showed yesterday, we produce a lot of trash. And so the number was higher, but the marketable daughter plants were not. So we did a 3D light map, and we saw that almost all of those daughter plants are growing under light compensation point. And if you don't know what is light compensation point, is the minimum amount of light that the leaf or a plant can receive, which can just survive. So it's the minimum amount. So we designed another experiment following these, and now I want to focus more because these are the new ones from last PIPCA. Yes.
I was just curious, did you actually tip out, or like try and establish and group any of the tips from this?
Yes, Dr. Bolt suggested in one of our meetings, and following that suggestion, we did that. And these daughter plants, the petioles are so long that we try to root them in the trays, and they do not even like, they cannot stay. And like, also I try to like, just for the sake of curiosity, put like a stick and like try to like hold them. And after like 2 weeks, the roots were like very small and fragile, while the other daughter plants, after 2 weeks, I have a like good amount of roots.
Even the ones with the 13.5 centimeters squared?
Yeah, the 13.5 was in the middle, but like we were shooting for like high planting density. So I tried 18 and it wasn't good. So the number of developed daughter plants are a lot higher, but the marketable ones are not.
Okay, I was just curious, because the other presentations kind of like, the fresh mass per.
So they look at the size difference of high quality. So here we have bigger and very small ones. Both of them are high quality, but the size is different. Here we have big, but they are really low quality, really long petioles, short leaves, really low chlorophyll, and they're not marketable. So yes, we followed up our experiments with another experiment on the effect of light distribution and adding intracanopy. So I want to just say that we did not provide more light for the intracanopy. We just changed that distribution because we didn't want to it's like more cost, more electricity. We just wanted to substitute some of the top light for the intracanopy. So with the same amount of light, we see how they will perform. So I don't want to go through all the background of intracanopy lighting because you all know strawberry and you don't want that. But in general, we had the top light, and also we provided intracanopy lighting. And for this intracanopy lighting to come up with the experiments, again, I don't want to go through the details of this, but we use the integrating sphere to have all the amount of photons coming out of the LED bars. So this is not PPFD, it is PPF. So here you can see the systems, top light, And also for the ones that they have intracanopy lighting, there is an intracanopy bar toward the other plants from again top. Our objectives was that to restore the quality decline of the daughter plants on their high planting density, and also to assess how this altered light distribution between top and runner canopy can impact the daughter plant production. So first quality, second yield. We hypothesized that partially substituting top light with intercanopy lighting at least in light compensation point, so at least around like 30 micromole in the intercanopy lighting will restore that daughter plant decline. Also, we hypothesize that it will also increase the total production because of more homogeneity in the like 3D planting area. We had actually 4 treatments. I decided to present 3 of them because the 4th one was a dynamic lighting and it's like really hard to present in this amount of time. So the first one, we had 100% top, which is the normal method that we were doing before. The second one, we had 90% top, 10% intercanopy during the production. But since we, in the first 6 weeks, so this is a 10-week period, in the first 6 weeks, we did not turn on the intercanopy lighting because basically we did not have a runner and we did not want to waste the photons. For the total production period, you can see the percentages are different. So, and the third one is 74% top, 26% intercanopy in the last week, which in the total production period is this amount.
What's the percentage against? Is that
The total amount. So if we have, for example,
wattage?
sorry,
Total wattage raise?
total PPF.
PP, okay. So
Total
that's-
PPF,
Okay,
80% of that
Okay.
there, for
Yeah.
example, 20 p.
okay.
Yeah. And the cultivars here are Monterey and Albion. So this experiment had 2 cultivars. So here you can see, this is a table, like simplified version that I put here. Each week, I know that this much is my top PPF, this much is my intercanopy. And until week 6, we did not have intercanopy lighting. After week 6, we turned on the intercanopy lighting. For all the experiments, the cumulative PPF is the same. So the same amount of light for all of them.
So the top lighting PPS 345, what's the canopy level PPFD?
PPFD was around like 400 something. Yeah, yeah. We also measure, but all the experiment is based on PPF.
Yeah, and the PPF based on per square meter or something?
It's not based on, it's not PPFD. It's the
I know.
total amount of,
No, but when you have 2 lights in the same space or 1 light, you know, you need a base, no? Even though PPF per fixture or PPF per.
PPF per growth
Or
area. Yes.
the total space.
The total is it. So we try to provide that PPF with like 3
Yeah.
bars. And based on that data from the integrating sphere, we know that at each dimming level,
Yeah.
each light provide how much amount of
And then
photons.
what's the space footprint of the growings?
It was like 1 foot to like 3 foot.
Oh, okay, so that's why you get high density
Yes.
PPFD.
Yeah,
because,
PPFD.
it's a square meter base.
Yes,
Got it.
yeah. So yeah, you can see a visualize of these, like how we distributed Albion and Monterey, and we repeated this experiment twice in 2 rounds and also twice at the same time. So there are like 2 replications at the same time too. And the planting density here is 19 plant per square meter. So we saw that the second hypothesis, production, was rejected. So the same amount of light, the same number of daughter plants. It's not gonna produce more daughter plants, both in Albion and Monterey. So this is, you can see in Albion is like 20 to 40 daughter plant per mother plant. In Monterey, we got 60 to 80 in 19 plant per square meter. When you
Over which time? So how many weeks?
10 weeks,
10
the table that I
Yeah,
showed,
yeah.
that one, yeah. So this, based on the per square meter, you can see Albion is around like 350 to 650. Monterey is around 1200 to $1,500 plants per square meter. Here you can see the quality. So this is 100% top, zero intracanopy, and these are those two. Both of them, the quality was fine, but this is a parameter that we decided to use if we want to present only one parameter for quality. What do we want to present? It's the amount of leaf area per petiole length. So you can see the leaf area here and petiole length, but the leaf area here and petiole length. It shows the compactness and it's the reverse of shade avoidance. So you can see in Albion, providing light, it increased the quality. In Monterey, also providing light increased the quality, but there was no difference in the amount of intracanopy lighting for increasing that quality. Here, you can see a real-time, like a photo. This is 74% top light, 26 intracanopy. You can kind of see the difference of the quality. All of those are like just petioles and really low quality other plants. I try to put another photo on the side so you can actually see what happened.
So do you think that in your Monterey experiment, if you gave more intercanopy lighting, do you think that because you had more runners on Monterey and got like more canopies, that the effect what you see there saw there?
That was one of the hypotheses that
Yeah.
because like I guess Monterey has a lot more,
Yeah.
that was we did not see that increase
Yeah.
further.
Okay.
So yes, this is what we saw. And we saw that like we can produce a lot of the other plants in a really compact area with providing intracanopy lighting, and they will be good quality other plants. So it's like 12 to 1,500 high quality other plants with intracanopy lighting. We were curious after the second round of our intracanopy lighting, and this curiosity, it's was from this table. So we talked and was like, we don't have any runner until week 6 because our plug plants are just like growing on the top and they want to get to that like canopy closure, and we have runners basically after week 6. What if we don't just like get rid of the mother plants, we keep them, we have that amount of like foliage on the top, and we have a second round of harvest. So you can see here the mother plant growth, and that 4-week in our intracanopy lighting was the like runner plant growth and that like area. So we had 4 chambers that we kept. So in 8 chambers that we harvested for, we kept 4 chambers. You can see Monterey, Albion, and we kept them for another cycle, we shoot for another 10 weeks, but I will tell you later that that didn't happen. So all of these plants, they went to different chambers, and we did that for the other cultivar too. So completely randomized. And actually, we had to harvest at week 7 because at week 7, they were all over the place. We couldn't even keep them for longer. So that 3 weeks, like until week 6, we had to here turn on the intracanopy after 3 weeks instead of after 6 weeks because we had runners earlier. And we had to harvest mother plants at 7 weeks. We saw that the number of the other plants dropped. So in the second, for all these light treatments that I don't want to go over the light treatment, they were basically that like 100% top and all of those in the first round, and the dynamic lighting, but regardless of the light treatment, for all of them, the number of the other plants dropped. And
Why here it dropped? Because would it be fair to say that it dropped because you keep them there for less time?
that answer is here.
Oh,
So we have 30% less time, but you can see 48% drop, 32, 48, 51, and you can see, so this is for Albion, that is for Monterey. So I put the percentages.
But you said You said the reason shortening is because there are so many plants already in the space. You
Sorry?
The reason you had to make a shorter period,
Mhm.
because you had more.
Like the runners were like until the ground.
Yeah.
When
so
they
that-
are like hit the ground,
Why it shows that?
so you can have less runners. You can have less runners or less like the other plants per runner. We should
The
go over.
internal length was longer then, or what happened?
Sorry, I should look at the data to see like actually we did not record the internal, but I guess the number of runners they were low. So they produced like 3-4 runners and those hit the
So
ground.
we had like less stolons.
Yes, the less stalls.
Okay,
Yeah.
less stolons.
The internal did not, yeah, at least visually did not
Right.
change because we harvest them based on yeah, we do harvest them based on area. I can provide that information later. Yeah, but the number of stolons were lower
Lower.
and the number of the other plants therefore was also lower. And the number of flowers per plant, so this is really interesting. You saw that in photoperiod, in a spectrum, we had like 10 to 15 flowers in the production period of 60 to 70 days. With environmental optimization that we had until this experiment, in this one, in the intracanopy lighting, we had like 2, 3 for Albion and 1 or 2 flower for the whole production period for Monterey. In the second round, it suddenly increased. Like we're all like removing flowers. One reason for it that it's my hypothesis is that during the harvest time of the intracanopy lighting experiment, during that like 4 or 5 days, because we had like 5,000 other plants, we dropped the temperature of the chamber because we didn't want plant to grow and the one that we harvest on Monday and the one that we harvest on like Wednesday, they are like different. So from 26, we dropped the temperature of the chamber to 15 to harvest those. And at that time, we did not even like plan to have this experiment. But later we talked about it like right after the harvest and we kept the plant. So I assume
And it was
that
16 hours, right? Yeah.
15, yes,
yeah.
yes, 16 or 15
Yeah.
degree centigrade. So I assume that initiated a lot of
But 2, 3 days.
the 5 days. Yeah.
So usually, I tend to think you have to have weeks of induction conditions.
Yeah, I don't know that.
Unless those are
If-
unique.
You suspect that maybe you had more flowers on the second half because you had larger plants, so you probably had more crowns to.
That's so it's either plant age, which is like they were like older and like larger crowns and like yes, or that environmental or combination of both.
This is, how many weeks of the second run?
So this first round is 10 weeks,
Yeah,
this is 7 weeks.
7 weeks, and
Yes.
then the flower is a bud stage or open flower?
We harvested the flower truss.
Yeah,
So
so
if-
already open.
Yes, like one truss that had like 3 flowers.
Yeah, so that means the fruit flower initials was about 4 weeks
Yeah.
prior.
Yes.
So that means you are still in second runnering condition, not the measurement time.
Mm.
So I think
When did you see the first flower?
something triggered, yeah. The size of the plants or.
Yeah.
Another thing is just the standard practice we do, when we don't see flowers in the production greenhouse, we mechanically prune,
Yeah.
and that stress somehow induce flowers. So I'm not sure if your harvesting action did that trigger, I don't know.
We just like pinch a flower with our nail, like we don't. Like after.
Stolen.
Oh, the stolen?
Yeah, I may
Yeah.
be totally wrong, but that's just associating with
Yeah,
what.
whatever the reason, like mechanical age, temperature, whatever,
Yeah.
it increased it
Yeah.
like. I was looking at the percentage, it was like 4,000%. So
Do you remember when you saw the first flowers of those plants?
I have the data. I have like weekly data, so I can just like look at them. It was like second week, I guess.
Second week.
Yeah.
Okay, yes, it will be in the first
Yeah,
plant, so.
Yeah, so this explain why you had fewer number of runners, right? Because your meristems occupied by flowers rather than
Yeah, we are also like now looking at a stolen flower ratio
Yeah.
in different treatments, and yeah, that will be a lot like different here.
Well, you have to
Yeah.
wrap it up.
Yeah, I'm done.
Okay.
So the number of stolen also, you can see that like in a lot of them, number of stolen dropped. And yeah, after all of these experiments, yesterday that the grower they were talking about like 20-30 per plant in 9 plant square meter, which I just did a like rough calculation. This is not a good chart, but like it's around like $300 plants. Now we are producing around like 1,200 to 1,500 in like higher plant density of 19 plant per square meter. I want to thank all of the students who were always helping me. And yes, if you have any questions. Yes.
So I may be crazy, but wanted to share. So. So the your inter lighting, it's the same quality as the top lighting,
Yes.
right? I was wondering if you can add far red in the inter lighting and make the Dora plants have flower initials. Do you think it has any benefit of doing that? So that's tiny materials already have flower initials. So that maybe branch ground, right? So that plant in a rooting condition.
Mhm.
And then instead of, the continuous terminal growth, branch crown induced, or do you get initially 2 crowns in earlier stage? I don't know.
So I know that even like both forward and also even longer photo period, it adds flower, and we saw flower on the daughter plants in those treatments.
Yeah, so
like in the first daughter plant, not the.
So that my idea is coming from your first experiment. Or
Yes.
whatever further experiment. So the top mother plant lighting, you don't want to put further, right?
But here,
But somehow if you can just target and then not reflection too much to the
So
mother
you're
plant.
talking for like tray plant production or that goes into.
Or even small flat.
so
Already 2 crowns.
there is a question: do you want flower in that like earlier stage?
Yeah, so that's what I'm saying, it's either flower or branch crown, so that's the benefit of having terminal bud into the
Yeah.
flower, right?
I
So I was thinking it's a sort of innovative
Yeah.
approach.
I'm also a little worried of if we have forest in that area, it will again elongate the daughter plants, which we don't want for sticking into the trays.
Right, yeah.
Yeah, I don't want any shade avoidance response in that area.
Like, oh, we have to wrap it up.
Thank you so much.